Thank you so much for coming, and it's a huge honor to be here. I have to say, so EEB didn't exist when I was here. That was starting to happen. This was 2000 to 2003. Obviously, as a division, you guys, as a department, you guys have done amazingly well. Continue to do so, and it's great to be back here in EEB. I want to start. Actually, before I start, I thank the people in my lab. A quick comment. My time as a Michigan fellow was really interesting. I came from a lab that uses microbes to study fundamental questions in ecology and evolutionary biology, but we didn't really do much real microbiology. And so I came here trying to make that transition and mostly failing during my time. First working on Vibrio cholerae with Victorita, and then learning about the microbiome, or that wasn't a word, the microbes that colonize the cystic fibrosis airway with John LaPuma. And I wound up spending another year learning about the microbiology of these organisms before I moved on to UNH. And that was a good thing. Working with Burkholderia, which I'll tell you about, is hard work. It's not as hard as coaching men's water polo here at the University of Michigan, which I did for 4 years. And in 2004, another good reason to leave, we won the national title and the Big Ten title. So even though I bleed green for one season per year, I wore the maize and blue and sang the songs, and so I'm with you in spirit. And I mention this because I see a Michigan men's water polo sweatshirt. Okay, so that said, and I still follow you guys, so keep up the good work. I really want to thank all the people who joined my lab at Pitt to help start it. So unfortunately, no one was able to make the move from New Hampshire to Pitt. And so in the last 2 years, this group has done a ton of great work. And I'm going to be, so we live down by the river. We're in a building a little bit away from the main medical campus and main biology campus. I'm in the medical school in the Department of Microbiology and Molecular Genetics. And we have this great space. And I'm going to be talking about work, primarily contributions from Nate Phillips, from Herman Traglia, who is a visiting postdoc, Alfonso Santos Lopez, Caroline Turner, a postdoc, joined our lab from the Lenski lab, and work from Dan Snyder and Isha Matre, another postdoc, and Chris Marshall, who arranged this photo and managed to avoid it. I'll really also talk about work from PhD students from my UNH lab, Stefan Poltak, Crystal Ellis, Kenny Flynn, Marcus Dillon, and Chuck Travers. Okay, so I wanted to be a field ecologist when I started, and the thing that really motivated me was originally was the Yellowstone fires. And what was stunning about that, I just visited, it's a beautiful place, and we saw conflagration changing the environment rapidly, and I thought, wow, this is a big change that we should be able to measure. How do we do that? And I started, and I went to college at Amherst College, and my advisor was Paul Ewald, and he said, doing field ecology and measuring, say, evolutionary ecology is great, but you're never going to get anything done as an undergraduate honors student. And maybe some of you have solved that problem, but instead he pushed me to this system where we studied evolution of a virus that infected gypsy moth caterpillars, and we evolved that virus to see how that influenced virulence. And we found that rapid cycling favored more rapid replicators, but that actually killed the insects when they were small, so they were less productive. And the fact that we could do an experiment and visualize a change in one trait that had obvious trade-offs had me hooked. We can do real experiments in time, and great, I'm impatient, let's do some more experiments. I moved to Rich's lab. You've probably seen figures like this many times. This is the famous long-term evolution experiment. Through 50,000 generations, they're now way out here at 68,000 generations, I think. That's where I lived. And I moved here, and I was looking for a more charismatic organism to come back to the question of the evolution of virulence, and I found out about the cystic fibrosis airway. And this is a situation where patients have this genetic disorder They have thick secretions because of this genetic defect in solute transport. It also has defects in ciliary function. People become chronically infected for years, during which evolution happens. And that was known phenotypically back then, although the functions were not known. And this is an example of Pseudomonas aeruginosa, where an early isolate from the lung airway produces less slime, less biofilm, and a late isolate from that same patient produces more slime. So that tends to be a common shift of increased biofilm production by varying mechanisms. Sometimes that produces more slime on plates, sometimes that produces effectively less slime on plates, but it changes their ability to adhere and form these sort of difficult to break up secretions. This is all exciting, but the question that was killing me is, what is the mechanism? Now, I was really fortunate to find one mutation in the long-term experiment. We found the first one basically by dumb luck. In a screen of losses of metabolic functions, we found a plate phenotype that was diagnostic of the first mutation. And it turned out to be one of the least significant mutations among the early set. It only had about a 1% benefit. The reason we detected it is that we could actually see it arising in the ancestor because it had a high mutation rate. We got lucky. So the question that I sought out to go after was, what do beneficial mutations look like, and what trait is under selection, particularly under conditions, under new environments, where those new environments might be disease. What do beneficial mutations look like, and what effect do they have on the population? Well, that question was becoming more relevant when we started looking at the epidemiology of patients who are infected by this other microbe that I'll spend most of the time talking about, the Burkholderia cepacia complex. Now, these are gram-negative beta proteobacteria, really charismatic microorganisms. They have 3 chromosomes. They can grow in a wide range of environments. They can grow in penicillin as a sole carbon source. Some of them can fix nitrogen. These microbes tend to be relatively late colonizers in the cystic fibrosis airway and are associated with particularly worse outcomes. And what these papers were showing is that when patients' sputum, when cultured, produced colony morphotypes that were heritable, that produced these non-mucoid types, we saw worse outcomes. And so this is a pure non-mucoid Kaplan-Meier curve for survival. This is the mixed outcome, and here is the non-mucoid outcome. This transition from mucoid to non-mucoid was clinically significant. So what's the molecular cause? What's the selective trait here? How do we look for it? And this also raised the question, could multiple bacterial traits or diversity itself be a target of selection? Okay, so we figured, well, we'll try and study something about biofilm evolution. And so I came to realize, partly by interacting with George O'Toole at Dartmouth Medical School in New Hampshire, that really we might have been studying bacteriology the wrong way for a century, because most microbes don't live in well-mixed environments. They live in aggregates or attached to surfaces in biofilms. And we don't know much about evolution in biofilms. And so although we can't easily recapitulate the cystic fibrosis airway in any way, there really is no animal model, we can just simply say, well, we'll subtract the host and we'll study biofilms themselves. Okay, so biofilms are difficult to reconstitute. In patients, they're sparsely sampled. We really still don't understand the demography of biofilms, so it's not just a binary state from sticking and swimming. In fact, it's a life cycle where there's attachment, assembly, dispersal to a new site, and then reattachment. How many cells actually make it through these processes? Where does division happen? This is something that our lab's really interested in right now. We need a model, and so we developed the simplest model we could come up with, which is essentially Rich Lenski's experiment with a plastic bead. Rather, we use test tubes. We put a plastic bead in the tube. We move the bead from test tube to test tube, where only the cells that stick to the plastic bead make it to the next test tube each day. Okay, so here's the cell. I put the bead in, bacteria attach to that bead. You take it out, move it to a new test tube, and there's an oppositely marked with a Sharpie marker bead in there where the cells have to attach, because we're going to transfer that the next time. Okay? And what's great about this is that you can archive it, and they have to reconstitute their complete community every day. So it solves a lot of problems. And so, basically, you have cells that have to attach, disperse, form a new biofilm each day. So one of my first PhD students, Stefan Poltak, took a clone of Burkholderia cenocepacia that was isolated from the soil, closely related to a lineage that had known to cause a lot of infections in North America, so genetically quite close to that lineage, came from the soil and ran an experiment. And in all replicate populations, diversity in the way of colony morphology arose quickly, right? So we have different colonies, and these colonies were conspicuous, heritable, and occurred in all populations. And so that made us wonder, well, why? Why are they happening over and over again? Why are these same types appearing? Might there actually be some niches here that these morphologies are reflective of? And so you can see that they form different amounts of biofilm. So these small colony variants really caught our eye. They are rugose. They are non-mucoid by diagnostic. They fit all the criteria in those papers about those that we were looking for in those patients with worse outcomes. We also have this ruffled type that produced more biofilm. This studded type is a bit of a generalist. It balances sticking and swimming, and it's numerically dominant in all populations. So we took one of the populations and took clones and labeled them, and with different fluorescent proteins. And what I'm showing you here is a Z stack. I hope you can see with the lights what I'm showing you is the studded type first, just only looking at the blue channel. You'll see it colonizes the exterior of the biofilm. We turn on the green channel for the ruffled, and you see it kind of fills in the gap and sticks to the plastic surface. And we turn on the red, and we don't see red so much as we see purple here. And that's because that red labeled wrinkly is sticking tightly to the plastic and forming these pillars, and the other guys are sticking on top of it. And so that's one way that these are diversifying, is by segregating space. And so here's the model that we came up with. You have the studded type on the exterior, you have the biofilm guys underneath. We also took these types and grew them by themselves and then asked whether the others could grow in each other's supernatant. And we're really surprised to find that the numerically dominant type was secreting things into the supernate, into the media, that could feed the two biofilm specialist types. They themselves couldn't feed each other, or one of themselves. And so we had really, just in a few months, the evolution of a kind of a primitive food web where you have a producer and two consumers that are paying them back, we thought, by providing substrate to stick to. So we thought, well, what are those ecological interactions? They could be competitive, or they could be facilitative. So we tested that across all the populations, again, by picking representative clones, growing each one of them by themselves, then growing them together. Now, if you know how much they grow, how much biofilm or number of cells they can produce attached to the bead by themselves, and you know what fraction they inhabit, their relative proportions in mixture, you can predict the mixed productivity. And what you can see is across all 6 populations, we saw more productivity in mixture than expected. So all becomes statistically synergistic. In some cases, up to 200% more productivity than expected. And we were able to calculate this using a really elegant equation that came from sort of plant community ecology that Michel Leroro and Andy Hector wrote to help analyze diversity productivity relationships in natural ecosystems. Okay, so we were really excited by this community ecology. And you remember the primary question that I'd asked. We wanted to know the mechanism. We had some inclination that some of these mutants might be relevant for at least some properties that we see in the CF airway. And then, mid-tenure for me, mid-assistant professor, the evolution detection machine was created, and that's the first high-throughput sequencer. And so there's the Illumina Genome Analyzer 2, producing 26 base pair reads, and by today's standards, not a lot of them. But even with that sort of primitive technology, we could sequence genomes and find the mutations under selection. We had no other way of doing that. And by the way, all the guesswork that almost anyone had ever done with a bacterial evolution experiment failed, which meant that we really didn't know what beneficial mutations looked like in the first place, and that the traits under selection were systems that weren't so obvious. And so even with that primitive technology, we could sequence populations and clones and put together a model of how this one population evolved. And so this is a Fisher-Muller diagram. This is mutation frequency over time. Again, blue for the generalist type that occupies most of the population, green for the ruffled type, red for the wrinkly type. Each of these M's are mutations, right? So this is the evolution of a haplotype now containing 5 mutations. And at this point, we find genotypes with this haplotype picking up another mutation that make a new ruffled and displacing the original one. And here we see another instance where a mutation is born on this haplotype and makes a new wrinkly. So even though we saw in all populations the rapid evolution of types that inhabited these niches, the specialists were continually being invaded and in some cases displaced. There's a lot to say here. I mean, what's great is that not only could we map dynamics, this is a sequenced genome, which is annotated. So we can ask about function, and we can actually think about microbiology, which is wonderful. What are the traits? So this trait, I'll point out, this M18 here is the mutation that's associated with this increase here. And this then goes back down, so the specialist becomes more common, and that goes back down when this mutation M7 happens and drives it back down. These turn out to be 2 different mutations in the same promoter upregulating the iron storage molecule bacterioferritin. So we never supplemented iron here. Most most microbes, but especially biofilm-forming bacteria, have a pretty high demand for iron. And so the specialist solved it one way by picking up one mutation, and then the generalist type picked up its own and knocked it back down. So we have tit-for-tat coevolution in this population over limiting iron. There's a number of mutations here, and we were really gratified to find that many of these mutations hit not only the same system, but even the same gene. So all of these blue boxes are mutations in the same gene that I'm going to spend a lot of time talking about now. And then these hit another cluster. And so if we take a look at all of the data from the 6 replicate populations that we sequenced and analyzed in this manner, half of these mutations hit one gene only once, but the other half hit the same gene multiple times. And the 2 that were hit most often are those in these blue and red boxes. This is a gene cluster that we've come to call WISP because it's very homologous to a cluster that had been annotated in Pseudomonas for the cause of wrinkly spreader morphologies. This is a pathway that Paul Rainey and Mike Travisano, Kerry Harwood, and others have studied in great detail. And then this gene, YCIR, which we named based on its name in E. coli, its homologous name in E. coli, that I'll tell you about now. Okay, so before I get into about YCIR in particular, I want to talk a little bit more about what we think the biofilm life cycle involves. So bacteria attach, initiate, and form an aggregate, and then they sort of commit to attachment and produce polysaccharide, form a more elaborate cluster, and then that biofilm becomes more elaborate, and at this point, there's plenty of physiological heterogeneity in the system. And some of these microbes in the middle are likely pretty starved for oxygen and nutrients, and that generates selective pressure. And some cells may then, for some reason, disperse, move on, and reattach. Now, biofilm production in most microbial systems, or many, maybe not most, but many, is associated with high levels of the second messenger molecule, cyclic di-GMP or cyclic diguanylate. And dispersal is triggered by the breakdown or low levels of that molecule. So these organisms likely have high levels of cyclic diGMP, and these organisms have low levels of cyclic diGMP. And in addition, these organisms are producing quorum-sensing molecules, and that is a density-dependent process. And so you can imagine that cells that are freely dispersed, which are secreting molecules in the external environment, are not perceiving many of them because they're being diluted. But as the cells become more concentrated, so too do those signals. And so they reach quorum at a lower threshold because of their density. And so these quorum-sensing processes can influence these strategies. Okay, so to introduce you now formally to YCIR, this is what it looked like, and these are all the mutations that we discovered in the initial long-term experiment. We've done many more experiments since, and it keeps coming up as a greatest hit. And this is really interesting because our genome has about 30 genes that look have attributes of this gene. Namely, they have domains that either synthesize or break down cyclic diGMP. So they have a cyclase domain and a phosphodiesterase domain. Again, 25 or 30 of these in our genome, and we never see mutations in those genes in our systems, only here, which is stunning. And then it has these sensor domains in the N-terminal region. It has this known sensor domain, and it has this domain of unknown function just prior to that. So something's going on there. So I had found out about this, and I'd met Chris Waters up at Michigan State, who's an expert in cyclic diguanylate and quite interested in evolution, does some evolution experiments himself, and then his colleague Matt Neidich, who's a great structural biologist at Rutgers. This has proven to be a really great collaboration because the structure of these domains is known, but this structure is not. Now, one reason why we think that this gene was under such strong selection is actually the gene that's next to it, which is RPFF, which produces a quorum-sensing molecule, a fatty acid quorum-sensing molecule, a relatively still understudied family of quorum-sensing molecules. This one here is called Burkholderia diffusible signaling factor, or cis-2-dodecanoic acid. And when we published our evolution study, a paper came right out afterwards from Leo Weberl's lab showing that BDSF binds this sensor domain, and also DENG, the DENG lag from Singapore. And they said they think it binds the sensor domain. So we said, well, right, this is really interesting. You have a synthase and a phosphodiesterase whose status is modulated by its sensing of a quorum-sensing molecule, effectively tipping the seesaw one way or another. And their model showed that when BDSF binds the sensor domain, it breaks down cyclic diGMP and promotes dispersal. Hence, this is a system that links sensing of the external environment with the decision to stick or swim in one gene. So we said, right, we got to figure out how this is working, and our data were not consistent with this model. And so in trying to solve this system, Matt Neidich's group and his student Evan Waldron crystallized and solved the structure of this domain of unknown function, which is passfold-like. I don't really understand it well either, but here is the model, which I love. They were really lucky. They actually moved to express this gene in a heterologous E. coli system, and also tried to co-express with other elements of the system, and found that this N-terminal domain of YCIR, domain of unknown function, is actually the RPFF inhibitory domain. It actually co-crystallized with the protein that makes the quorum sensing molecule, the gene next door. So how they think it works is that in the absence of BDSF, these two folds are locked together. But in the presence of BDSF, it opens them up, and then it binds RPFF, which shuts down the production of BDSF, the quorum sensing molecule. itself. So it's a feedback inhibition loop, and it's modulating the sticker swim sulfate decision. So okay, this is supposed to be an EEB seminar, Vaughn. How does this work? Well, we have some predictions. We have mutations that evolved in these different domains, and they have structural predicted functions. One of the genes, one of the mutations, A106P, is right in between these two folds. And so the prediction is that it prevents interaction between the inhibitory domain and the pass domain, and that's going to make this quorum sensing signal blind, which means it's going to be ignoring the signal to disperse, and it's going to promote biofilm, and it's also going to make cyclic di-GMP levels high. And we had this other mutation which had all these phenotypes associated with it, which led us to hypothesize, oh, it must somehow be stimulating the diguanylate cyclase activity because it's in that particular region of the protein. And so with Chris's help, we measured cyclic di-GMP levels, and we find that's in fact the case. We see high levels of cyclic di-GMP in the Y355D mutant, in the A106P mutant, and when you knock out the enzyme that produces the quorum sensing molecule that's supposed to be triggering dispersal. Okay, they also produce alone different ecologies, right? So here is A106P, it's producing these bigger clumps. Here is Y355D, it's producing a more uniform structure. You put them together, and you can see in some cases they're forming aggregates, and this is sort of their Z stack, so you're looking at them from the side. And so that's consistent with our original model. Individual mutations in this gene are generating the initial adaptive radiation. I'm not showing you WISP here, which is in filling in the blanks. And this has broad implications because this gene is very well conserved. It's very closely related to a gene that Matt Chapman's lab knows well from his strain. When you knock out YCIR in E. coli, it influences its ability to produce biofilm in a variety of phenotypes, biofilm mediated by Curli, Fimbriae production. And the production of these molecules, BDSF-related molecules, are common. And in fact, when we co-express these enzymes in E. coli, we found that these Burkholderia genes bound another molecule just like this, lauric acid. So this implies that this signaling pathway is broad, and it could help us understand how to modulate biofilm-associated infections through these messenger molecules. So this is exciting. This all came from the lab, and we then said, well, what's going on out there? Just to point out that, to summarize, adaptive diversification in this system was driven by mutations in two loci, yielding 2 to 3 coexisting ecological strategies whose combined actions were synergistic, if you remember back to that original mixed versus monoculture plot. So I mentioned we were interested in what's going on right out there, and I'll provide 2 examples. And I'll only talk briefly about WISP here, but this is probably the best example. These are caused by single mutations that activate the WISP cluster. And they produce this diagnostic high biofilm phenotype. Dan Wozniak's lab at Ohio State has been studying these rugose small colony variants in Pseudomonas aeruginosa for quite some time. And when I was talking about this in sort of this environment, he said, you know, I wonder if these types of morphologies that come out of our study of burn models might be driven by the same forces. So his postdoc, Aaron Gloag, with Chris Marshall in my lab, picked a bunch of these small colony variants from these burn wound models. These are sadly pigs. One of the best models of chronic wounds. And these morphotypes arise very early on, but they don't fix. Right? So they reach about 10% frequency, and when they're there, the wound healing is delayed. The molecular cause, the sole cause of these variants is mutations in this WISP pathway in homologous sites that evolve in our test tube model. So that implies that a lot of early selection in attaching to a new environment, particularly for opportunistic pathogens, not professional pathogens, those that are adapting from another environment and learning to stick to something else, evolve by picking up mutations that don't fix, that generate diversity and improve attachment per se. And it doesn't matter what may not matter so much what that tissue or substrate is. And so the broader implication is that evolution can do genetics for us in environments that we care about. And so I had been reaching out to ask, well, can we see these dynamics of evolution from isolates, serial isolates from infections? Because if we find them and we find those mutations occurring in traits over and over again, that could point to pathways that could be therapeutically useful. And I've already suggested that this dispersal signaling pathway through YCR might be one of them. So even in advance of moving to Pitt, I met Yohei Doi, who's an amazing clinician who's sort of evolution-minded. And he pointed me at first to one of the major industries of the University of Pittsburgh Medical Center, and that's transplant. Transplant patients are inherently immunocompromised, and they're subject to these opportunistic infections during which drugs are used, all of them, and they sometimes fail. So one of the latest drugs is ceftolozane tazobactam, or Zerbaxa. It's a combination beta-lactam, beta-lactamase inhibitor. And some patients were having good outcomes and others, treatment was failing, and they got longitudinal isolates. We sequenced, and we found mutations in particular regions. Not surprisingly, in the region where the beta-lactamase inhibitor hits the beta-lactamase produced by the microbe. So this isn't too surprising. These sites hadn't been known, so we learned a little more about the enzyme. At a minimum, this promotes some diagnostics. This infection was a bit more interesting and kind of led us down a new road. So I had met Jason Roche at a meeting, and he said, hey, we have this broad, this is a St. Jude's Medical Children's Research Hospital, and most of those patients have cancer or are primary blood disorders, and they're subject to a range of opportunistic infections. This infant picked up a bloodstream infection of vancomycin-resistant enterococcus that failed all treatment. So it turned out to be likely associated with a contaminated central venous catheter. She was originally treated with vancomycin, then gentamicin and daptomycin, then linezolid, gentamicin, daptomycin, linezolid again. I can't pronounce that antibiotic, and nothing worked. But the good news is she survived because she was given a granulocyte transfusion from her parent that reconstituted her immune system. And this is important because it shows that the pathway to drug resistance, whatever it was conditional upon the lack of the immune system. Right? So that the evolution of drug resistance depended on being immune compromised. And so St. Jude has immense resources for sequencing, not only for cancer exomes, but also for microbiology. So we sequenced the bejesus out of every isolate, expecting to find lots, and we found one mutation, just one. A single mutation in the gene REL-A, interestingly, hits this system that governs the stringent response, production of an alarmone, another messenger that tells cells whether to hunker down or not. Dysregulation of that system by its partner, SPO-T, is it is the most common mutation in the Lenski long-term lines. Weird. But this single mutation had a biofilm-dependent phenotype. By that I mean it altered expression of hundreds of genes, but for a subset of genes, it changed their direction of expression, going up or down in the shift from the planktonic to biofilm lifestyle. And so what I'm showing you now are drug resistance profiles in biofilms grown by these organisms. This organism is actually fairly susceptible to drugs in planktonic conditions, but when grown in a biofilm, it's resistant to all of these drugs that were treated. And so it raises this question then, was the original target of selection acting on this microbe attachment to the catheter via a biofilm or drug in the first place. In some sense, it doesn't matter, right? But you can see how these two functions are interrelated. Now, okay, so these are sort of opportunistic, short-term ways to find single targets of infection. Many of you know, right, that patients who have CF, the life expectancy has gone from 30 to 50, since I started studying it, which is fabulous. The bad news is that the duration of these infections is extended, right? And so microbes can inhabit these patients' lungs for decades. And so what I'm showing you quickly now is a story about sort of a biography of one infection, this one patient, who was selected by my colleague in Portugal, Leonildo Moreira, because it had this pattern of switching from mucoid in red to non-mucoid in black and back and forth again. Remember, this is this pattern that was associated with worse outcomes. Fortunately not for this patient. She's still alive. And so we said, well, what are the mechanisms and how is this population evolved? And so we sequenced all the genomes, we found all the SNPs, and we find a fairly linear increase in SNPs across the population over time. But when we build a phylogeny, we find multiple lineages. And if we go back to the original plot and color those isolates by their different lineages, you see that at any given time, there are multiple coexisting lineages, and they last together for years. And this is certainly an underestimate. Follow-up studies of studies like this, where you sample more and more frequently, find more and more coexisting lineages, partly because they're geographically isolated in different regions of the lung, but also partly because they may be physically interacting with one another. And also, because these are independent clades, this allows us to ask again, do we see evidence of parallelism? So if we ask, do we see mutations occurring in different clades hitting the same traits? So in some sense, you can view each clade as a semi-independent evolution experiment within this patient. And so we asked, well, how many genes get hit more than once? And we found 10. We found a significant overrepresentation of genes that were hit multiple times across this one infection. Some hit as many as 4 times, and one of them that really caught our eye was this one, the sensor hexidine kinase FixL, named FixL because it's involved in nitrogen fixation. So, environmental microbiology and host microbe interactions have lots in common, even across kingdoms. In this case, we think this gene is being reappropriated for a new function relevant to the CF lung. And the reason we're really excited interested in it, is because this turned out to be the gene that was mutated most times in a study of many patients who were infected by a point source clone of another Burkholderia species at Boston Children's Hospital. This is beautiful work done by Roy Kashoni, Greg Priebe, led by Tammy Lieberman. She's now got her own lab. This gene was hit 17 times among this patient studied here. So really strong evidence of selection at this trait. We've now collaborated with a bunch of folks around the world and found mutations here, again, never taking over the population in the gene fix that are associated with worse outcomes in those patients. So we found them, here in Michigan, associated with a particular clone called Midwest clone, in Boston, in Vancouver, in Portugal, and most recently in Argentina, associated with another medical supply associated epidemic. This is another trait which is both diagnostic and potentially a target that restores susceptibility. So we can study evolution in action in infections using the lens of genomics. These evolved mutations point to selective targets. And I think a big opportunity is that there are thousands of genomes with clinical data available. One of the biggest threats facing humanity now is the evolution of drug resistance. I'm fortunate now to have a big project where we're studying the evolution of drug resistance on a pretty big scale, both in biofilm and planktonic conditions. I have some slides here that I'm going to skip just to say that we can now use lots of evolution experiments as a forward genetic screen to find out how bacteria adapt to different drug concentrations, both in test tubes, in planktonic and biofilm conditions, and in mice under varying immune states, and find qualitatively different dynamics. Hopefully some other time I can give you a lot more detail about that. I'm skipping that now because I want to get to the most important work that our lab does. So sorry about this. Okay, this question, what do beneficial mutations look like and what do beneficial mutants look like, used to be inaccessible. But now we can sequence and find them. And we can design experiments where selection is strong and evolution may produce phenotypes rapidly, which provides an opportunity to teach evolution better. And so we've worked hard really for the last 10 years to try to translate these experiments to the classroom by building a system which we call Evolving STEM. It is the same biofilm model with a safe microbe, Pseudomonas fluorescens. At the outset, students, these are 9th graders, doing the experiments in the place of their typical evolution curriculum. We'll inoculate a tube with 2 markers, 2 versions of that clone, lac, plus, and minus, that make 2 different colors, blue and white. And after 5 days of transfer from Monday to Friday, they'll see the evolution at the community level, a lot more biofilm, and then they'll plate, and they come in on Monday, and they see morphologies that are different. And we've been trying to explain this in the context of the story that basically every high school student that gets any evolution learns, and that's the evolution of diversity in Darwin's finches. This is an adaptive radiation into different functional types, and then they can take these if the school invests more and phenotype them and learn, well, what is the equivalent trait to beak size here? Is it more biofilm? Where do they grow? And if they spend even more time, they can do inquiry. So a wonderful school and set of teachers partnered with us back in 2014 and divided their classes in 2. One class got our curriculum, and the other class got the standard curriculum, and then they switched. And so we then measured learning with a common metric, and we found that those that got our curriculum on that standard test did way better, statistically better, than those that got the standard curriculum. But the good news is that when they switched it, the other group caught up. And this was reproducible. We did it one more time. So this is super exciting. You learn better. They also did much better basically on the open answer questions. They could talk more about evolution in their answers than simply on the multiple choice questions that we gave them. That was hard. What's easier to do is now to survey what are your sentiments about science, technology, engineering, and mathematics before and after this curriculum. Now I know that this is early days, and I know that this is only after the sort of an immediate follow-up. There's a lot more to do. But we were thrilled by these initial results. This is a meta-analysis now of students from 4 schools where we ask a variety of Likert scale questions, sort of from strongly agree to strongly disagree. And I'm just showing you the % favorable responses, strongly agree or agree, broken down by gender, pre and post. And what I want to emphasize are a few points that are broadly interesting and also gender specific. So this one right here, I enjoy using technology. Boys love technology. These are, again, 9th graders. They go from 84% favorable to 93% favorable. And the girls, sort of in the middle, meh. But if we ask the next question, I am good at using technology. There is no change for the boys, but there's a 20% increase for the girls. Using technology helps me learn science, mathematics, or engineering. Goes up 20% for the girls, 10% for the boys. I will make it into a good college and major in an area needed for STEM. That's cut off. Goes up 14% for girls. Goes down slightly for the boys. I would like to have a career that involves STEM. Goes up for both genders. How can we not do this? This is easy, and we're building a kit that can be distributed here. So a lot of the work that we've been doing is in test tubes, but we've recently developed a plate-based model, which we'd like to distribute to as many schools as possible, and nobody in Michigan's doing it. So I would love to help. We currently have schools in Pennsylvania, in Pittsburgh area, New Hampshire, Montana, and California doing this. And I'd like to spread this as far and wide as possible. You can find out more about this at evolvingstem.org. And before I go to the grand conclusions, I want to go back to the big question now. Maybe useful for everybody, what do beneficial mutations look like? And so I thought about this and say, and the only thing I can really come up with is that when bacterial populations, big populations, are exposed to a new environment under strong selection, they have to move fast and stuff gets broken. And some of those mutations break traits that are not normally broken in nature. And so what I can say is these represent short-term solutions and hence are not found in nature except under similar short-sighted evolution. So like other cystic fibrosis infections, other drug treatments. They affect genes that are often redundant, but they are not structural. They tend to be regulators. They tend to be non-synonymous mutations or partial loss of function mutations in regulators. They influence pathways that are of intermediate connectivity and centrality. And the thing that's driving us crazy, and I was talking to George about this today, is they're not as pleiotropic as we would have thought. So we don't understand this. We would think that hitting these genes that are conserved in nature would be pleiotropic in our lab conditions, but evidently we're not measuring them. And that probably is because the environment's a heck of a lot more complicated than we give it credit for, and there's really no parasites and not a lot of microbe-microbe interaction in the models that we're studying. So I want to thank you all for your attention, and I want to thank all the folks of my prior lab that helped get us here. And thank you again for your attention. The ones that no longer... Yeah. Yes, we have. And we've also shifted the conditions of the experiment to sort of rinse the bead vigorously and only keep the guys. So we will shift the frequencies of these types, but they're still there. And it leads me to believe they're forming aggregates too. So if you look at a fully grown population, a lot of the cells actually that probably were forming aggregates in the bead wind up getting knocked off. And I should say these tubes are not static, they're on roller drums. Beads are bouncing into one another and stuff's falling off. So, I mean, the population is the population, right? You're not going to go back and find wild type after a while. But yeah, it's gone. Yeah, it doesn't. But we have done the experiment, and that's sort of how we solved how WISP works, is we've gone from the mutant and selected back planktonically for wild type trait, and we find suppressors, right? And then we do the same thing again, and then we do the same thing again, and you can basically decompose the pieces of that system that's producing that WISP trait. That works, and eventually you run out of solutions, and it doesn't work anymore, and that's sort of also gratifying, I think. Yeah, so these are pretty specific adaptations, and they come at cost, and some of the mutations themselves are not revertible, although we have observed reversion of the WISP mutation in a few rare instances, showing that, at least in that case, that's pleiotropic for that biofilm planktonic trade-off. Does that answer